Stat 135, Lecture 24. Great job on a tough midterm. I've shown here the grade cutoffs. Last time we were talking about goodness of fit. That's section 9.5. You'll notice that it seems like I might be jumping around a bit, but I'm doing the chi-square tests all together, so we'll be doing also section 13.3 and 13.4, test of homogeneity and independence. But the idea about goodness of fit is that you have one categorical variable that has certain multinomial distribution. We'll cell probabilities P1 to Pm. Theta can have k components, and our key result is that the chi-squared Pearson test statistic is a chi-squared of dimension m-1 minus k. Today we're going to be doing test of homogeneity and independence. Just to put all of these different chi-square tests together to show you how they all fit, we have for one categorical variable the degrees of freedom, the goodness of fit and test of homogeneity. Goodness of fit asks the question, does the categorical variable have the claimed multivariate distribution? Test of homogeneity is about two or more subgroups of a population and asking whether those all share the same multivariate distribution. Alternatively, you could have two categorical variables and test whether those two categorical variables are independent. So test of homogeneity, just to start with an example, a study was done comparing frequencies of a particular allele in a sample of diabetes and non-diabetics. So we've divided our population into two groups, diabetics and non-diabetics, and for each group there are two categories. Let's just say type BB and type capital B, capital B. And so we can look at the relative frequencies of each of those categories. So for example, 12 divided by 51, 39 divided over 51, that is that's a binomial distribution there. And we have two categories for non-diabetics, 4 over 52 and 49 over 52 are those probabilities, and that's also binomial. And you could ask whether those binomial distributions are the same. So now let's just do some theory here. So let's say that we have, instead of 2, we have j independent I cell multinomials. So let's say that I is 2 and j is 3 for point of illustration. Our null is going to be j, the j multinomials are all the same, and the alternative is that they're not all the same. So our null box now consists of these binomials, pi 1, pi 2. This is for diabetics, and we're assuming that that's the same as for non-diabetics, and let's make a third category somewhat diabetic. So all three, we're assuming in our null box those distributions are the same, and the sample size for each of those are, we'll call that n.1, n.2, and n.3. So the dot refers to the sum of the counts n11 and n21. So I've shown here the observed counts and their marginal counts, and you can see then that n.1 means that I'm adding up n11 and n21. Similarly, n.2 and n.3. So we're going to need to estimate pi 1 So since we don't know what pi 1 is, we're going to estimate it. So pi 1 would be, well, so we have three different subpopulations: the black, the purple, and the green. So what we're going to do is we're just going to add up all of the counts for the N11, N12, N13, call that N1 dot, and divide that by the total count. So that's our estimators. And now our expected counts are going to be, so since each of the cells in our observed box is binomial, which has expectation NP, so for the top left, our sample size is n.1, and our p would be pi 1, but we don't know pi 1, so we substitute pi 1 hat, and so we get n.1 and 1 dot over n. And similarly for the other cells. So we have our observed, we have our expected counts. I just want to make a remark that you could ask, what if we divide the population according to allele type instead of according to whether or not you have diabetes? So the fact is that you're going to get the same observed and expected tables. Let's just check that. So if we were to divide our population according to the I categories, so in this case allele types, so if we then we would have two subpopulations, type big B little b and type big B. That's shown in blue and purple here, and we would have three categories for each of those subpopulations: diabetics, non-diabetics, and somewhat diabetic. So then our samples are N1 dot and N2 dot, because we're summing over the allele types, and pi 1 hat would be n dot 1 over n, and similarly for pi 2 hat and pi 3 hat. So this will give us our expected table, and you can notice that this is exactly the same table as we had before. In fact, our observed and our expected tables are exactly the same as above. So it does not matter what you call your subpopulations and the categories of those subpopulations. It all works out the same. But let's go back to, right, so you could compute the chi-squared statistic now very easily from the observed and expected tables. We need to figure out the degrees of freedom. So chi-squared is the dimension of omega minus the dimension of little omega. Little omega is, here we have one multinomial, because our null box is that there's just one, with I minus 1 free parameters, hence the dimension of omega naught is I minus 1. And capital Omega, we have j multinomials in general with I minus 1 free parameters per multinomial, hence j times I minus 1. So the degrees of freedom, you can check the algebra, it works out to be I minus 1, j minus 1. We can then compute the chi-squared statistic as usual and get this expression. So next we're going to do test of independence. So this is a test whether two categorical variables are independent. So we'll just, to motivate this, modify the problem that we were doing above. We have the same data, but now the question is the relationship between whether you have diabetes and allele type related, right? Is there a relationship between whether you have diabetes and whether you have allele type? So if you make a scatter plot, you'll see that it'll look something like this, And if you were to draw the best line fitting through your data, because most of the points are having type big B, your line, the best fitting line would essentially go through those 2 points, and it would have slope 0. So that shows you that you have no dependence, or that the two variables are independent. You can compare that with, for example, if you had a plot like I'm showed on the right here, this would clearly have a negative slope best fitting line, a negative correlation between the 2 variables. Okay, so now some theory here. So we have one J cell multinomial, one I cell multinomial, and so I've written out that probability means, independence means that pi ij is pi I pi j. So our null is that the 2 random variables are independent. The alternative is that they're dependent. And so my null box looks like this. So here I have I as 2 and j as 3. So I have a total of 6 cells. So everyone in the population has one of 6, it fits in one of 6 categories. And you could then take a sample of size n, and you would get these counts, and you would get the following expected counts. So pi 1 hat and pi 1 hat for I and for j are as calculated above here, and you'll see that you get exactly the same expected table that we did get for test of homogeneity. So we're going to get exactly the same chi-square test statistic, and we'll show next time that this also has degrees of freedom I minus 1, j minus 1. So the test of homogeneity and the test of independence are exactly equivalent. They just correspond to different experimental designs. Have a great day.